Okay, we're going to go ahead and we're going to look at the phylum Zygomycota first. These are commonly referred to as zygomycetes or zygomycetes, but the true phylum name is Zygomycota. So if I ask you to give me the name of the phylum, that's what I would be looking for, is Zygomycota. So these tend to be very fast-growing molds. They can be parasitic and they can be symbiotic depending on what they're on. But I would guess that the one that you're probably most familiar with is what this is showing you up here in the left corner of figure 3112. And this is very common. It's called the black bread mold. So if you leave your bread in the pantry for too long, especially if it doesn't have a lot of a preservative in it, then this will come sooner rather than later. But eventually it will come in all bread species. They are able to reproduce themselves both asexually and sexually. So they've kind of combined both stages of the life cycle here in this one figure. And this is basically kind of nice because it summarizes what's going to happen and shows the structures for both sexual and asexual reproduction in one slide. And you'll want to pay attention because you'll be looking at both of these in your lab when you get to that part of the class. So guys, if we look at the we take a close-up, right? This is mycelium. It's growing on the outside of the bread now because it's running out of food and it needs to reproduce itself. Okay, so the mycelium has been in the bread the whole time. It's in the bread that you eat. You just don't see it. It doesn't do anything to you. You're only going to notice it when it comes from the inside out, when it's ready to sporulate, because again, it's going to be running out of food at some point. So it needs to spread to other things in your pantry or a different part of the loaf of the bread, for example. So if we take that and we look at this right here and we look at it over here, right? So you can see it's kind of this like little type of anchor with individual hypha that are coming up here with these little black circles at the top. And they kind of almost look like little lollipops, if you will. Well, that right here is this over here. There's the little anchor. So these are individual hyphal strands, mycelium, and then individual hyphal strands coming up here, getting ready then to make spores so that the fungus can spread itself asexually. So when these spores then are made in the sporangium, you should know that term, sporangium. Remember, it means holder of spores, right? They're going to make thousands upon thousands of spores. Those spores will be released and they'll make more mycelium. It'll make more fruiting bodies or asexual fruiting bodies to make more spores so that they can be released, and then more mycelium comes. So it's just a repetitive cycle here. What they want to show you here is that coming from this spore that has cracked open, there's a hyphal strand. It's going to start to branch off. You can see that there's the little red nuclei in there. This would be an example of aseptate hypha. It branches, it grows, makes mycelium into this. This is this. So when you are looking at this under the scope, for example, when you're completing your lab, you can see that there's an individual hypha here. And at the top, this is going to be the sporangium. And then all these little brown circles here are going to be the spores that are encased then on the top of the sporangium. This will crack open and eventually release the spores. The spores will make more hypha, which makes mycelium, so on and so forth. So that's pretty simple. So this again is the asexual side of things for Zygomycota. Now, if and when sexual reproduction needs to occur, this is what's going to happen. You're going to get spores. The spores are going to come from two different fungi, but members of the same species. So maybe from that sporangium and that sporangium, okay? And they're going to be released. And one's going to germinate, if you will, and start to grow a hyphal strand here. The same thing's going to happen here, but what they want you to notice is that they're showing you the nuclei are different colors, just so that you can understand that this is the species A. This is also species A, but they're two different members of that species. They're going to happen to call this one mating type negative, this one will be mating type positive instead of male and female. So there's a hyphal strand, here's a hyphal strand. The hypha is going to grow a projection out towards the left. This one's going to grow a projection out towards the right. These hyphal tips will eventually fuse. And if you remember, that's going to give us then what is called plasmogamy. So plasmogamy now, I have then a cell with different nuclei, the red and the blue. Okay? And it can stay that way for quite a long time. If you remember, it's the environment that's going to give the cue that says, okay, karyogamy can happen. So this here, let me back up for just a second. The plasmogamy is hetero or dikaryotic. And once those different nuclei are in the same cytoplasm, this particular organism in the phylum Zygomycota is going to make a hard spherical structure around this area to protect then those nuclei. And that is going to be this guy here. Okay, this is going to be called a zygosporangium. So sporangium is holder. This time it's holding the zygote because the zygote comes from sexual reproduction. Notice this is the hyphal strand from the negative mating type. This is the hyphal strand from the positive mating or plus mating type. So now the environment says, okay, those two dikaryotic nuclei can go ahead and fuse. They do so. Now we're to our diploid state. So if the red and blue here mate, or I'm sorry, not mate, but fuse, the red and the blue fuse, the red and the blue fuse. That's all happening within the zygosporangium. I'm going to end up with 3 diploid nuclei just because that's what the picture is showing. So in the zygosporangium, remember that meiosis has to occur. So these diploid nuclei then are going to go through meiosis. And coming out of then that zygosporangium is going to be a hypha with the sporangium giving different spores. The only difference between this sporangium here coming out of the zygosporangium and this sporangium here coming off of the mycelium is that these spores are all identical to one another because that's asexual reproduction, while these spores here are all different because it's sexual reproduction. Okay, so like this picture over here, this is another micrograph of a slide that you guys will be looking at. There's the hypha from the plus strain. Here's the hypha from the minus strain. That's your zygosporangium, and it's housing then the zygotes inside, the diploid zygotes inside. This here is giving many spores right off the bat. This has to go through meiosis before it can give many spores. Okay. So again, just kind of another slide showing the same things here. This is going to be the asexual micrograph that you guys will look at in your lab. Here's the hypha. Here's the top of it. So this bulbous part in the hypha is the sporangium. These are the spores. Asexual. Here, hyphal strand, plasmogamy has just occurred there. But over here, hyphal strand, here's your zygosporangium with the zygospores inside. And that is pretty indicative of the group Zygomycota. Okay. So I just want to make sure I'm just looking at their notes here. So for A, phylum Zygomycota, for number 2, it says asexual reproductive structure, sporangium. I want you to remember that the sporangium just means holder of spores. And then for number 3, sexual reproductive structure is the zygosporangium. That's the holder of the zygote. So if it says zygote, then you should automatically know that that has something then that has to do with sexual reproduction. Okay, our next group then are going to be in the phylum Ascomycota. Ascomycetes or Ascomycetes then is the common name for this group. These guys are a little bit more common probably than the Zygomycota. They live pretty much everywhere and anywhere with a few exceptions. They're not really found in extreme environments, but you can find them living in marine type of environments, freshwater, and then all over on the terrestrial type of an environment. Most of these guys are going to be multicellular, like you can see here. that they're showing in figure 3116 from your textbook. But this is going to be the one group, or where the one group is located in phylum Ascomycota, and those are going to be your yeasts, and the yeasts are going to be then the only organisms that are unicellular within the kingdom. And maybe you've heard of yeast if you've baked bread and things like that. Okay, it's a rising agent. Okay, so if you look here at this figure, it's the same thing we've been looking at. You've got your asexual reproductive stage over here. You've got your sexual reproductive stage over there. And you've got haploid, dikaryotic or heterokaryotic, and then diploid back to haploid. So nothing changes here, guys, except the structures themselves. Okay, so a couple of terms that I want to point out for asexual reproduction here in the Ascomycota. The sporangium is actually called a kinidiophore. So I want you to realize that the kinidiophore here is the same exact thing as the sporangium. It just has a different name, and it looks different than any of the other sporangia that you've looked at up to this point. So kinidiophore, okay, this means holder. Holder of what? Well, conidio is for conidia. So this is a holder of conidia. Conidia are the specific name of the asexual spores here. So I no longer just refer to them as spores in this group. They are now specifically referred to as conidia, and that term is going to be right up here. So just so I can back up for just a second, the kinidiophore is the same as a sporangium. The spores here are called conidia. Okay? Now, for the sexual reproductive structures over here, that's where I want to spend a little bit more time. We're going to have this guy here. So the phylum Ascomycota, these guys are commonly referred to as the cup fungi. And the cup fungi makes sense because if you look at the structure right here, this thing called the ascocarp, that is the cup fungus. And this is very indicative of what the sexual fruiting body looks like for the group Ascomycota. Hence, cup fungi. So the ascocarp is the sexual fruiting body. It is lined on the inside with these long tubular structures. Ascus is singular, asci is plural. So there's one long ascus, there's a second one, a third, a fourth, a fifth, a sixth, a seventh, and there's 8 of them there, okay? And they are filled then with ascospores. And before they break open, they're always going to have 8, 1, 2, 3, 4, 5, 6, 7, 8 ascospores. So let me back up and talk about this again. The ascocarp is the sexual fruiting body. If it is an edible type, you would be eating the ascocarp and everything inside of it. Lining the inside of the ascocarp are asci, these individual tubes. There's one, there's another one, there's another one, and there's another one. Inside each ascus will be 8 ascospores. And so this story is the same. The ascospores are released from the ascocarp when they are mature. One germinates, the other germinates. You get a red and a blue, plus and minus, right? They're going to fuse then over here. They're going to fuse their hyphal tips. We're going to have plasmogamy, red and blue. So 2 different haploid nuclei. That's dikaryotic. Environment says, okay, you can go ahead and fuse. They do. Now you have a diploid nucleus, and inside this structure here, which is the ascus, meiosis is going to occur. You're going to get haploid spores that are called ascospores that are mature over here, that are these brown structures over there. So 8 ascospores. Okay, for this group, you guys are going to look at both asexual examples and sexual examples. So let me go through that with you. So for the asexual representatives in your lab, you guys are going to be looking at Penicillium and then another species called Aspergillus. So individual hypha, making this little kind of broom structure. This right here would be the kinidiophore, this guy right here. So here's my conidiophore with the individual spores called conidia at the tip. Penicillium means tiny artist's brush. So the first person to have identified this species of ascomycete thought that the conidiophore, this structure right here, looked like a little tiny artist's brush. So that's where the name comes from. This one is small and it's hard to see, so the micrographs aren't going to show up as well as the aspergillus might. But this is the same thing. This is a single hypha with the bulb here. That's the conidiophore with the little lines of conidia coming off of it. So this kind of looks like peacock feathers to me, if you will. This structure and the conidia is exactly the equivalent as this conidiophore and its conidia, but they're just from different genre, and so they look different, okay? But they have the exact same function, and they would both be represented here in the asexual side of things. Okay, Penicillium conidiophore with conidia, Aspergillus conidiophore with its respective conidia. Okay, now let's look at what this would look like from the sexual side of things. So here is a species of an ascomycete called Phyla, and you can see this looks like a cup, doesn't? That is the cup fungus. This is the ascocarp. That's the sexual fruiting body. We can't see it with the naked eye. It just looks like it's kind of empty inside of there, but it's not empty at all. If you were to slice this guy and take a peek at it, here would be maybe just like one of the sides right here, making the curvature of the ascocarp. This is all the hypha, the mycelium that came together to make the ascocarp. And then closer to the top of the surface, you're going to have your asci. You can't really make them out very well, but then each ascus is going to have 8 ascospores, which are the red structures here. So if we take a peek at this and we magnify that, this is what you have. So this is all the hypha and the mycelium making the ascocarp. This here is one ascus with the ascospores. Okay, here is another ascus with the ascospores. Now, once in a while, you'll be able to actually make out the 8 ascospores, but you have to remember when they make the slide, some of this tissue gets crushed and some of the ascospores are released, or you just can't see them in the view. But each ascus would, upon maturity, have 8 ascospores. So you guys will also be looking at this here then in your lab. Okay. So hopefully that makes sense. Once again, if you're super confused, please send me an email or Zoom me, and I'd be happy to talk about it with you. Okay, here are some other examples of ascomycota, just so you can see that they don't all necessarily look like the cup fungi that they're named after. This right here is a morchella. It looks like a mushroom, but it's not a true mushroom because we haven't got to those yet. But that would be, in fact, a sexual fruiting body. And each one of these little pores right here would be lined with asci with 8 ascospores. Okay, so here's the little cup parts. It's not just one cup, it's many cups at the top of that sexual fruiting body. Over here, this is what's called a truffle or a morel. This is something that they hunt for and harvest in France. They use pigs to sniff these out. So if you've ever had shavings of truffle on a dish at a very fancy, like French restaurant, for example, they're going to be quite expensive. They are a couple $1,000s for just an ounce or so of these guys. If you've ever cooked with truffle oil, it's a very expensive type of oil that comes from these. I've never had one, but they are a delicacy. And again, you have to go with pigs that sniff these out under the leaf litter. But again, it doesn't look very much like this close cousin, but if you were to cut that guy open, it's going to be lined with asci with 8 ascospores in each ascus. So this right here is going to be the ascocarp. So that's the cup portion of the fungus. Okay, our last group, which you'll be happy to know for the phylum Basidiomycota, they only have a known sexual stage. That doesn't mean that asexual stages don't exist, but nobody has got to see one. And so all we can talk about then is the sexual stage here. So there's a little bit less that you have to remember. So for this phylum, this is the one I would guess that you're probably most familiar with. These are going to be things like your mushrooms and the puffballs, shelf fungi, and the like. Okay, so we're going to go through the life cycle here, and then I'm going to proceed to show you some examples. Okay, so equivalent then to the ascocarp that we saw in Ascomycota, instead of having a cup fungus or a truffle or a morel, you have a mushroom. And so the mushroom itself is the basidiocarp. Okay, so you want to think of the basidiocarp as the sexual fruiting body. It is going to have little gills on the underside of the top of the fungus. Maybe you guys have seen that, like when you're, I don't know, cooking mushrooms or eating mushrooms. And on those little gills, so let me get my pointer back here, on these little lines here, that's what you're looking at here. Okay, so they're highly folded for an increase in surface area. If we look at just one of the areas there, then what's lining the gills are these little club-like structures, these little billy club-like structures called basidia. And those basidia are going to go through karyogamy. You're going to get your diploid nuclei. Those nuclei are going to go through meiosis, so the story is the same. And then in each of the basidium, then you're going to get, eventually from here to here, you're going to get these 4 little basidiospores. So this is a basidium, this structure right here, holding 4 basidiospores. So if you were to look at it under the microscope, here it is. This is the basidium with 4 basidiospores. And those are, again, are going to be sexual spores. So this is similar to the ascocarp, but this is a basidiocarp this time, making then a container, if you will, to hold your sexual spores, but instead of having them inside, the basidium is going to have them on the outside. And instead of having 8 here, you're only going to have 4. Those basidiospores are going to be hanging down on the underside of the cap of the mushroom, so that when wind currents come and those spores are mature, they're going to take those basidiospores right off of the basidia. They're going to germinate after they land, and you're going to have that hypha germinate. You're going to have that hypha germinate. Notice 2 members of the same species, mating type the negative, mating type of the positive. You're going to have then the 2 hyphal strands come together. You're going to get plasmogamy. Here's our dikaryotic mycelium. So none of those nuclei, right, they're just in the same cytoplasm at this point. They haven't fused. That will happen only over here when the environment says go ahead and do so. Meiosis occurs, and then you end up with 4 haploid basidiospores on each basidium that line the gills that are on the underside of the mushroom cap. Okay. Here are some examples of mushrooms. Here is just the top of it, and here's the little stalk portion of it. This is a coprinus mushroom, and this is the mushroom that Disney a lot of times will use when they make their movies and things like that. It's kind of a happy mushroom, right? It's a pretty mushroom. There are species in this genre that are completely fine to eat, while there are others that are completely toxic. So again, unless you know what you're doing, do not just try to sample mushrooms. This is giving you then a peek underneath the cap of a mushroom, and those are all the gills, and those gills, as you can imagine, are just lined with millions upon millions of basidia, and then therefore there are 4 basidiospores. This down here, this is what's called a fairy ring, and so remember the mycelium is usually somewhere where you can't see it, so it's underground, under the soil here, it's under the soil there, and only when this mushroom needs to reproduce itself will it pop up this basidiocarp, i.e. the mushroom. So this fungus that's in the ground all over this field here has decided that it is time to reproduce, and so it is going to then pop up all of these mushrooms simultaneously, and it makes a perfect ring almost called a fairy ring, like I mentioned earlier, and it's going to release billions upon billions of spores. They will be taken out from under the cap and they'll be spread maybe over here to another part of the field, and then they'll germinate, they'll grow hypha, which will then grow into masses of mycelium. Here are some other examples. This is a puffball, and you can see that it releases a smoke-like substance, and so every couple of minutes or so it'll give a puff, and that, what you guys are seeing, that's going to be millions upon millions of spores. Not all mushrooms look the same. This is going to be called a shelf fungus. This one is growing outside of a log of a tree that's fallen down, and so what this fungus is doing is it's actually going to decompose then the wood of that dead tree. And then this one here looks so different than any of the others. This is called a maiden veil fungus, and you can see that it doesn't have the typical cap and gills. It's actually very fragile and lace looking, okay, but they don't smell very good. Okay, I just want to mention you've got 2 YouTube links here that you might want to take a peek at. One of them is showing just a puffball. Somebody is pressing on a puffball, and then you have all of the little puffs of smoke of spores that you can see coming out of it. And then the other one is a video showing the release of spores from a mushroom that has opened, and it's a pretty cool little video. And the one of the mushroom releasing its spores is here, and that one's narrated. This one I believe is the puffball, and it's very short, a matter of, I don't know, 30 seconds or something, just so you can see the actual spores coming out of it. Okay. Again, I just wanted to make sure that you remember that there are fungi that are not so good, and these ones, actually I've talked about, I mentioned all of them. These are all going to be, I believe, at least these 2 for sure are going to be in the phylum Basidiomycota. Okay, this will help you with your lab, and I've given some space in your notes at the top of page 2 to draw this. So this is what you need to know. You need to know that the top of the basidiocarp, or the mushroom, is called a cap, or the technical term is the pileus. On the underside are going to be the gills. You do not need to know the word lamelle, just know gills, okay? Remember that the gills are lined with basidiocarps that have basidia with 4 basidiospores, okay? You need to know that this portion right here, this is called the stem, or the stipe or stape. You can spell it with an I or an A there. And that is all you need to know. So again, you need to know the cap, pileus, you need to know the name of the gills, and you need to know stem or stipe. You need to realize that this entire structure is the sexual fruiting body called the basidiocarp, and it is made up of mycelium. Okay, just some more examples. All right, so let me just back up for just a second. If we take one of these mushrooms, some of them actually, like this coprinus species, it liquefies itself. So once it opens its basidiocarp, the cap or the pileus here, releases its spores, and then it's done. It's done its job, right? Because that's the only thing it's there for, is to reproduce, and it will basically turn into like a black ink and then totally disappear. But if you take a cut of the cap or the pileus and you look at it, you turn it upside down after you cut it. This is going to be the center where the stipe was connected to the cap. You're going to see that these are the gills, right, that are lining the inside of the underside of the cap or the pileus. And if we take a peek at that, here's a gill. These little kind of platform structures, if you will, these are all the basidia, and then these round structures are all of the basidiospores. And you can see from this micrograph here that each gill is lined on both sides with basidia and basidiospores. So this right here could be this area here, and then this right here would be the neighboring gill there, and then another gill and another gill and another gill. So you can imagine how these basidiocarps can make up to millions and millions of spores per structure. Okay, to finish up then, we have our lichens, and lichens are mutualistic associations between the fungus, which is going to be all of the kind of yellowy stuff here. That's all fungal mycelium, usually from the phylum Ascomycota. And then embedded and entwined within the mycelium and these individual hyphal strands are these little green circles, and those are going to be then your green algae, usually from the group Chlorophyta. Okay, so here's what's going on. You've got then the hyphal strands of the mycelium from the fungus. This is the gray area here, surrounding then and wrapping up the green individual or unicellular green algae. And what these guys do is they live in close, intimate relationships, because what's happening is, if you can notice here, if you were to look at this underneath the microscope, all of the green algal cells are close to the top of the lichen where they can get sunlight. And so they're autotrophic, as you should know, and they're making sugars, and they're going to make a little bit of extra sugar, and they're going to give some of that extra sugar then to the fungal hypha. And in exchange for that, then the fungal hypha is giving basically the algae a protected place to live and making sure that they are positioned as best as they can be so that they can get light at all times. They are important in primary succession. So when you take it down to, remember we talked about like the rock, and then the rock has to be broken down to make soil, and then secondary succession. Well, the lichens are one of those things because they release really strong acids that can actually break down rocks and start to have the formation of soil occur. We don't really see very many of these because, unfortunately, they are really, really sensitive to high levels of air pollution. So obviously you're not going to see them in Bakersfield, but if you go up into Tehachapi, you'll see them there where the air is a little bit cleaner. If you go hiking out in like Sequoia and things like that, you'll see them there. Okay, what you'll need to know is that, first of all, again, that they're made up of two organisms, a fungus usually from the phylum Ascomycota, and an algae from the group Chlorophyta, your green algae, okay? And they come in three different forms, so three body types: foliose, fruticose, and crustose. So I've given you some space there in your notes to draw them. You will also be doing this in the lab, and it is up to you to know when I show you a picture of one of these, what the name of the body is. So this one here, if I just start over here on the left, it's kind of a shrub-like structure, right? So all of this that you're seeing right here is all the fungal mycelium. The algal cells, because they're so small, they are microscopic, so we can't see those, but you would know that they are going to be in here intertwined with all of the mycelium, and that is called fruticose. Okay, if I come over here and I look at this one, the fungal hypha is it's not as stringy, it's more flat and broad, more leaf-like, and so they've named that the foliose lichen. So shrub-like is fruticose, leaf-like is going to be foliose. And then sometimes you'll see they look very different than either the fruticose or the foliose. And this type of lichen here, if I can get my cursor back, the orange is again, the fungal mycelium. It would have those microscopic algal cells in it. And this is called crustose. It's encrusting. That means that it is basically glued itself so tightly, releasing acids that it cements itself to the rock. So you almost have to go and chisel it off. You can't just peel it off like you could either of the foliose or the fruticose. Okay, so you'll be responsible for drawing those three body types of a lichen in your lab, and you need to know, again, the name of each of the body forms, okay, just by sight. Okay, that concludes then our lecture for chapter 31. Like with the plants, guys, what I'm going to tell you is that life cycle is the thing that you're going to have to go back over, I would imagine, a few times just to make sure that you really get it down, so that you can take then the different phyla, Zygomycota, Ascomycota, and Basidiomycota, and you can overlay, if you will, then their specific life cycles, their specific structures, and the name of the structures. Remember, the structures all do the same thing, but fun for you, they all have different names and they look a little different, so it's just more to study. Okay, that concludes the lecture. Thank you for listening. If you have any questions, please do not hesitate to email me. Thank you.